Hello and Assalamu alaikum everyone. I would begin by thanking the scientific committee of the 19th MMIDSP annual conference for inviting me to give this talk. I am Syed Hani Abdi from Aga Khan University, and the topic I'm going to present today is landscape of HIV in Pakistan with focus on drug resistance mutations. So as a brief introduction, HIV, the human immunodeficiency virus, is a lentivirus and approximately has a 9.8 KB genome, so is a relatively small virus. A single-stranded positive sense envelope RNA virus, and by the virtue of having the RNA genome, it's really capable of mutating fairly quickly in response to host pressures as well as the drug resistance pressure. It primarily infects the CD4 T cells, but as the infection progresses into chronic and the late chronic phases, it can infect many other cells, such as the dendritic cells, the immune cells, the osteoclasts, osteoblasts, and many other cells. Currently, there are no cure, no vaccines, but a very effective treatment is available, which is antiretroviral treatment or a combination of several drugs, which is known as highly active antiretroviral treatment. When we talk about HIV, it is really important to consider the types and the subtypes of this virus. HIV is broadly divided into two types, HIV-1 and HIV-2. HIV-2 is limited to certain geographical regions such as Kenya and other countries, Brazil and etc. And not many cases are reported from this subtype from different parts of the world. HIV-1 is the main pandemic subtype and is actually responsible for all HIV cases throughout the world. Based on the sequence classification, HIV-1 is further divided into three main groups, the O or the outlier group, N or the new group, or M and the main group. N and O are limited to very few countries and very few infections have been reported from these two groups so far. However, group M is the main group which is responsible for almost all HIV cases throughout the world. HIV group M is very diverse and it is further subdivided into pure subtypes and several recombinant forms. The pure subtypes are from A to D, F to H, J to K, and so far over 120 different circulatory recombinant forms are actually reported from this group, and more have been seen are being seen each year. This map shows the distribution of different HIV subtypes in different parts of the world. So you would appreciate that, for example, subtype B is common in North America, in South America, and Europe, and also in most part of the world. In Africa is C, and in certain regions we have subtype A. The situation in Asia is quite interesting and quite diverse. So for example, we have subtype B and C as the main subtypes in our neighboring countries such as China and India. But in Pakistan, we have subtype A as the main epidemic type. Uh, Throughout the Asia, subtype O1AE, which is a recombinant form, is the major epidemic form. So this is the representation of the HIV genome. I've already have told you that HIV has a 9.8 KB genome and it has 9 different genes. GAG, POL, and envelope are the structural genes. TAT and REV are the regulatory genes and they take part in several processes that are important for the virus life cycle. While VPU, VIF, and VPR are the accessory genes and they play a crucial role in evading the host response against the virus. So the inhibitors or the drugs that are available so far, they either target envelope as a fusion inhibitor, or most of them that we have so far and are used even within our country, they target the POL protein of the virus. So this shows the HIV reverse transcriptase protein, which is part of the POL protein. Reverse transcriptase converts the viral RNA into DNA, and this actually shows the catalytic site of the RT. So you would see that this is the viral RNA, and this is where the incoming nucleotide is. And this is how the catalytic site of the enzyme will interact with the viral genome and convert it from RNA into DNA. So this is the viral RT protein, and we will see where the NNRTI or drug resistance mutations are strategically located. So the drug etravirine, which is an NRTI inhibitor, would interact with the viral RT proteins over here and would block the incoming nucleotide. So this is how it's supposed to inhibit RT. And you will see the NNRTI resistance mutations, how they are very nicely strategically located around the drug, where the drug binding site is, and they would nullify the effect of the NNRTI inhibitor. So let's look at another example. This is the viral protease enzyme, which is an essential enzyme in the HIV life cycle. It cleaves the GAG-POL polyprotein and matures both the POL and the GAG proteins. So if you look at the PI inhibitor, which is the loponevir drug, it actually sits at the protease catalytic site and block the activity of the enzyme. And you would appreciate how both the major and the minor mutations are strategically located to nullify or reduce the impact of this inhibitor. So major mutations are those which are sufficient to nullify the effect of the drug, while the minor mutations, they are the compensatory mutation, so they will make up or compensate for any loss in fitness because of the major mutations, or they would help the major mutations in exerting their full effects. So the idea of the slide is actually to appreciate how the drug resistance mutations, they strategically emerge at the site near to the inhibitors and would either nullify or actually suppress the effect. Before we go ahead and talk more about HIV drug resistance, I think it is important to understand how the mutant variants they emerge, and they become dominant in individuals and then can also be observed at population level. So let's assume that this is an HIV positive individual who is on ART therapy, and because of that, he may have the wild type virus as well as the mutant viruses. So if this individual transmits the virus further into HIV negative or positive but ARP naive individuals, in these individuals, not only just the wild type, but especially the mutant virus will be selected. So when they are put on the therapy, these they will be at an inherent disadvantage because the resistance virus is already there in the system, and it will buffer the effect of the antiretroviral drugs. So if these mutations are selected and further transmission goes on, so this become transmitted resistance mutations and then can be observed at different population level, right? So, and this is not a hypothetical situation. This is also observed in many cases. We have sequence in Pakistan where certain drug resistance mutations are observed at population level. They might have been selected during transmission, or they might have evolved while the patient was on the ART inhibitors. In any of the cases, certain of these mutations are getting selected within our Pakistani individuals, and they have become amplified at a population level. So when we sequence many patients from different part of the countries, these mutations are frequently observed. So let's look at some of the projected models that have been done to look at the HIV resistance to treatment responses. So on my left, there is a case with a drug which has a low genetic barrier to resistance, meaning any mutation can make the drug ineffective. And on my right, there's a drug which has high genetic barrier to resistance, meaning certain mutations would not affect the major activity of the drug. So if you look at the CD4 card on the viral mRNA, you will see that because there is a low barrier to resistance, the CD4 count is expected to decline soon, and there will be incomplete viral mRNA suppression. So there will be a continuous expression of viral RNA within the host. In case of high genetic resistance in case of drugs which have high barrier to resistance, you will see that there will be a decline in CD4 count, but gradually, right? However, if you look at the viral RNA, uh, it would be at the patient would exhibit a lower viremia as compared to the individuals who are on drugs which has low genetic barrier to resistance. This is an important model which looks at an individual which has received a pre-selected mutation. So a mutation which was transmitted to this individual who is ART9. So in the very first view, you'll see a rapid decline of CD4 count and rapid sharp increase in the HIV RNA, and which will lead to rapid suppression or decline of the CD4 count. But the viral load from the circulation would be reduced, but viral will soon enter the latent phase because it will start seeding all the important cells. So, and these will happen fairly quickly as compared to drugs which have higher resistance barrier, and they are actually would take months for this effect to happen, right? So it's important to keep an eye on individuals who have received pre-treatment, who have, sorry, received a transmitted drug resistance mutations, and these mutations can really affect the treatment outcome by virtue of a lower CD4 count in individuals and a sharp increase in the viremia. So before I move on to discussing the results from our lab, I think as an important slide, we could look at some of the challenges related to ARV surveillance resistance research actually. So foremost is genotypic variability. So there's no perfect definition for genotype specific mutation even to date. And we talked about that there are over 120 different recombinant forms. So it's very, very hard to classify which mutation is specific to which genotype. And an individual who is affected by a particular genotype or a particular newer recombinant form, for example, can be at a particular disadvantage and the drugs may not work. I'm not saying that the drugs will not work. There's a chance that the newer recombinant forms may resist the effect of the drug, but especially when there is no definition of what genotype specific mutation is. The second challenge is that the mutations are hard to classify. So certain mutations, they occur in the absence of drugs, for example, in response to the immune selection pressures, and they have a dual effect on the drug resistance as well. So this becomes a really important challenge to characterize which drugs are they emerge in presence of the drugs, and which mutations emerge in the presence of the drugs, and which mutation emerge in the absence of the drugs. The third challenge is the study differences. So there, if you look at a comparison of all the studies from different regions of the world, there's quite a difference. There is consensus on mutations, but at the same time, there's also difference in respect to region, time, study population. So we often ignore the population genetics or selection pressures, but this play a very important role in deciding how the virus will evolve and what kind of mutation it will harbor in its genome. And finally, the burden of HIV drug resistance mutation is very, very high. So there are approximately 300 studies published within a span of 5 years looking at genotypic resistance in ART naive patients only. So I'm not talking about those patients who are on treatment, but patients who haven't started the therapy yet. So the burden of drug resistance mutation is quite high. So these challenges are really important to consider while planning or executing a surveillance, our surveillance study, or considering a programmatic approach to implement our resistance surveillance at a genomic level to go with the clinical monitoring of the patient. So somebody, if still is wondering why this topic is so important, so there's some of the facts from the studies are this that especially in the resource limited regions which have the higher burden of art resistance mutation, approximately 25% of the patients who are receiving the first line art therapy, they develop virological failure within 1 year. So virological failure is inability to suppress viral load beyond 1,000 copies per ml after 3 to 6 months of the treatment. So very high burden of the patients, they develop virological failure, especially in the resource limiting setting. The drug resistance mutations are responsible for 50 to 90% of the virological failures actually. So it's a very high burden of virological failure is attributed to the drug resistance mutation. The regimens that we use in the resource limited countries may differ from those that are used in the well resourced countries. Also important to consider are the subtype differences. So maybe due to the genotypic differences that we completely do not know, the treatment may vary between one region to another. Uh, some of the important considerations, especially in the resource limiting countries, including Pakistan, is that the patients are monitored infrequently with respect to viral loads, with respect to especially with respect to drug resistance mutations. So viral load may vary from center to center or country or province to province, but I'm almost sure that not too many places would frequently monitor the emergence of drug resistance mutations. The second line therapy is chosen without doing the genotypic resistance testing. So many of the mutations are assumed and they are assumed based on the genotype B. So let's say if you have AE or O2AG or A as a genotype, circulating genotype, you may end up in cases where the drugs are may not be working. Um, and the first line therapy is chosen without the looking for transmitted drug resistance mutation. So we do not have a very nice classification of whether the ART, naive, and heart experienced patients, what kind of baseline mutations do they have. And I've actually talked about that. I want to re-emphasize is that the subtype variations are completely ignored. So we really do not know what is the, like I said earlier, that there's no perfect definition of what is a genotype specific mutation, but we also assume a lot of mutations based on subtype G. However, it might differ in different genotypes, especially those which are newly emerged or newly have recombined to form a newer variant. So with this brief background, I'm going to now switch gears and talk about the landscape of HIV in Pakistan and emphasis on drug resistance mutation. I'm going to start with presenting a fact sheet for Pakistan. Then we will going to look at the subtype diversity within the country, focusing just on HIV. Then we are going to look at the phylogenetic models and how different high-risk groups are communicating with each other. And finally, I'm going to present some of the data on the drug resistance mutations. So this is HIV fact sheets related to Pakistan. The data I've took from is from UNAIDS and NACP website. So currently there are 160,000, more than 160,000 people who are living with HIV. Uh, the sad part or the concerning part is that the HIV prevalence in the country is really, really high. So if you consider different high risk groups, the prevalence varies from 11 to 37%, and which makes Pakistan as one of those 25 countries which has prevalence of HIV more than 25%, especially in the IDU community. And you see the proportion to positivity ratio. The proportion of individuals who are receiving treatment is just around 15,000 to maybe 20,000 individuals, and not of all of them are drug adherent actually. So a huge number of patients are actually are not on any of the ART therapy. And the most concerning part is the sharp rise in the number of new infections. So between 2010 to 2021, I would say there's approximately a 45 to 47% increase in the newer HIV cases, which is really, really alarming. So while from in other part of the world, a decline in HIV cases have been observed, but in regions such as former Soviet Union countries or in Pakistan, um, the number of newer HIV cases are increasing. So situation in Pakistan with respect to HIV is quite alarming. So this figure, based on the sequences that have been deposited into HIV databases, this is the HIV subtype diversity in Pakistan. So you will see there are quite a few subtypes that are circulating within Pakistan. The major ones are subtype A, which actually is responsible for approximately 65.9% of the cases, and rapidly subtype O2AG, which is a CRF, circulatory recombinant form, this is rapidly taking over. So now the newer sequences that we are sequencing or amplifying from the HIV patients, most of them would belong to O2AG and not A. So it might happen that we are experiencing a real-time shift from one subtype to another. But A, so as we speak, is still the major subtype, followed by O2AG, but not, but there are several other subtypes such as O2A1, B, C, G, and other recombinant forms. So this is an important slide for us, and this is the original research data that we have generated in just a period of 5 years. And this shows some of the new circulatory recombinant forms or unique recombinant forms that we have identified in Pakistan. So for those of you who don't know that, a URF or a unique recombinant form is a recombinant form of HIV which is sequenced from a single individual. When this URF is sequenced from more than 3 to 4 individuals, they are actually then classified as a CRF. So from Punjab, we have sequenced a CRF 56 CPX. We have also sequenced URF A1, which is a unique recombinant form from an individual from Punjab. From Karachi, we isolated sequence and amplified in sequence URFDG. And from the outbreak investigation of HIV outbreak in Larkana 2019, we identified CRF02AG in 15 individuals. So, which actually makes a very important case that the Pakistani epidemic is much complex and much more diverse than we previously expect. And if we, let's say, implement a full-on sequencing program in Pakistan, so we might be able to identify various CRFs and URFs from different patients. The point of concern, especially with these CRFs, is all of them were multi-drug resistant, especially against the NRTIs and PIs. So these are not just that they are recombining and emerging as a newer variant of HIV, they're also multi-drug resistant viruses. So if they're further transmitted to individuals, the situation is only going to get concerning, because transmission of these viruses would lead to large-scale emergence of drug resistance in different individuals. So from here, I'm going to switch gears a bit and look at or talk about HIV transmissions in Pakistan. So this is another arena which is really underexplored and also fairly complicated and diverse actually. So on one hand, so the thing you see on a slide, this figure you are seeing on the slide is actually a phylogenetic tree. What a phylogenetic tree basically does, it infers the relationship between different viral sequences. So where it actually looks whether the sequences are connected, they cluster together. So are they coming from the same source? Are they actually going in the same direction? And what is the relationship between different clusters? So on this tree, you will see sequences from IDUs, intravenous drug uses. You will see sequence from high risk group that we call as men who have sex with men, MSMs, and then also several of the families, the spouses and the kids of the MSMs actually. So in this slide, I would like to emphasize a very important point. So you will see several mixing points where IDUs are actually here and here. They are mixing together with the MSM family. So and then the MSMs are clustering or grouping together with the spouses and the kids. Sequences, HIV sequences, which we have amplified from the kids and spouses of MSMs, they tend to orient in a family specific manner. So if if I want to simplify this tree into a model, so the way the transmission is happening in Pakistan is that IDU is primarily transmitting to MSMs, and obviously MSMs, especially those who are IDU MSMs, they are transmitting back to IDU. So there's a close relationship, and a close bidirectional transmission of HIV between IDUs and MSMs. Then for to save to not to get affected by marginalization and to have a face for society, MSNs tend to marry and then to have kids. So we have 8 different families as an example where virus got transmitted from the father who was also an MSM to spouses and then to the kids actually. So like I said, it's very much underexplored. We do not know what's happening in different parts of the country, but the data we have indicates that the high risk groups are also interacting in, let's say, in regions from Karachi, and the epidemic is now bridging from high risk groups to low risk groups, including women and children. So another important point of concern and very a point to support the case for large scale molecular epidemiological studies on HIV. So, moving away from to a different region, which is our work in Larkana, and you will see the situation is quite different. So here you'll see 3 different kind of sequences. The black ones are the reference sequences, which are non-Pakistani, so you can kind of ignore those for now. But the main relationship is between the Pakistani PWIDs or IDUs and other high-risk groups and the Larkana sequences. So Larkana sequences are shown in yellow color, other high-risk groups are shown in blue color. So you'll see that while most of the sequences would form a discrete cluster, but there's some mixing between Larkana and IDU sequences. This might indicate that, especially in regions of Rotadero in Larkana, the virus was present within the high-risk communities and then got transmitted through poor infection control practices to low-risk populations such as children, right? But for other subtypes, this is a very interesting situation. So we talked about how bridging is happening, but there's also very discrete clusters. So you'll see that a cluster solely comprising of the Larkana outbreak sequences. So if you put these three phylogenetic trees together, the situation that would emerge is that the HIV, especially in Larkana region, was there in the high-risk communities. It got transmitted nosocomially into children, and it was not just acquired in 2019 where the outbreak happened. This has been happening for quite a number of years actually. So the virus there's evidence of ongoing transmissions within the community, and so HIV silently and rapidly is spreading throughout the community. So it's really important to do a comprehensive molecular epidemiological studies to look especially at the transmissions of HIV and then channel our public health interventions to stop these transmissions. The talk is also focused on drug resistance mutations. So in the next 3 slides, I'm going to talk about different NRTIs, NNRTIs, and PI resistance mutations. I'm going to acknowledge that we are currently not typing the integrase inhibitors, and we have just established the protocol, and hopefully in a matter of few months we're also going to start looking at the presence of integrase resistance mutations, if any, especially with the introduction of DTG as a first line regimen. So this data that I'm going to present is just focused on RT and the PI inhibitors. So some of the major mutations such as N103K or E138E, they are highly prevalent, which are effective NRTI resistance mutations and can nullify the effect of NRTI inhibitors. They are prevalent in not just the experienced group shown in red, but also in the naive group. They have a very high prevalence. So most of the patients you sequence would exhibit the E138A minor mutation or K103NTE major mutation. So this mutation is sufficient to abrogate the effect of NRTIs. E138E works as a compensatory mutation, and together they work synergistically to nullify or substantially reduce the effects of NRTIs, and they are highly prevalent in our patients. So moving on, now we're going to look at the NRTI resistance mutations, and you'll see that certain of these mutations, for example, VL15Y, uh, which uh, which affects the efficacy of abacavir, is prevalent in not just the drug experienced patient, but also the drug naive patient. Similarly, we have M184P and K65R. They are also major mutations, and they can take away the entire first line RT based first line therapy. So they nullify the effect of ABC, TDF, or D4T, and if they are co-inhibited in a patient or co-amplified in a patient, the entire first line is actually gone. One interesting thing is, and this is also um, um, important to note, that alone Y115V might not be that important, but together with K65 E or R, it can take away the entire NRTL line. And these are observed in most of the patients that we have amplified in our studies. And these are the results for the protease inhibitor resistance, and you'll see that important mutations K20R, which renders nelfenor ineffective, is prevalent in both drug resistance and both drug experienced and drug naive patients. So I already told you that we haven't looked at integrase inhibitors yet. We are going to do that in the next few months. But the data for protease inhibitors and the reverse transcriptase inhibitors that we have so far indicates that several of the good percentage, I would say, of the patients are resistant against the RTIs and the PI inhibitors. So a comprehensive testing of those patients, especially of the ART naive patients before starting the therapy, is really, really important. Especially with the introduction of DTG, it's really important to go for integrase testing so we do not lead to a situation where the DTG resistance mutation also becomes common, because from the literature we have reports now that some of the viruses from Africa and other parts of the world where DTG resistance is also observed. So this is just a recap slide based on the effect of mutations on different classes of drugs. We have PIs, we have NRTIs, and we have NNRTIs, and and this actually shows what percentage of drugs are affecting what classes of the what kind of drug resistance mutations are affecting what classes of drugs. So you see that most of the mutations that we observe in our patients or Pakistani people living with HIV, they are mostly resistant so far against NRTIs and NRTIs, but also few of the protease inhibitors. So up to 50 to 60% of those mutations, they target the ATV and the LPV, PI inhibitors. But the prevalence against the NRTIs and NRTIs are quite high. So we hope to get the data on the integrase inhibitor soon, and then we can see whether the resistance is already there or whether the DTG is safe so far. So we do not have the data, so it's very hard to comment what is happening with DTG so far. But as far as NRTIs concerned, the resistance is quite high, and also against the PI inhibitor. This is a very important slide, especially for those who are involved in the molecular biology research. But this looks at the baseline maps, what is happening at the viral genome, and this measures the entropy, which actually is the propensity to acquire newer mutations. So these are the regions which I have highlighted in pink color. These are the regions where drug resistance mutations would technically evolve. And you'll see that the Pakistani viruses, they have a very high entropy in those regions. So the possibility of acquiring newer mutations is very, very high in the Pakistani strains that are circulating so far. The other important point that caught our attention was look at the maps between the naive and experience and the biological failure. So the patterns of entropy that we have for the biological failure, it does not resemble to that of experience, but that of naive actually. So in summary, on a non-technical term, the viruses that the individuals who are drug naive, the kind of strains they've acquired, the genomic signatures look exactly like those of biological failures, which is very, very alarming, because if there's multiple pre-existing mutations, especially in the sites shown in pink here that are the drug resistant sites, So it means that the current inhibitors might not be so effective in controlling those individuals. But this is like not all the 160,000 patients. This is cohort-based studies, but the signals we are getting are alarming actually. So again, a very important point to sequence the patients before starting the therapy, because if the genomic signatures matches of the virological failure, the resistance and virological failure is almost deemed to happen. And I cannot emphasize enough how important it is to continuously monitor the patients for not especially before starting the therapy and also while the patient is on therapy. So with this, I'm going to conclude my talk, and I hope I was able to make a case about the landscape of HIV in Pakistan, especially with in terms of subtype diversity, different high-risk group, transmission between different high-risk groups, and then the drug resistance mutations. So I would like to conclude with these points that there's higher prevalence of against NRTI and NRTI resistance mutation, especially in our naive and experienced individuals alike. So in order of prevalence, the most restriction, resistance was directed against NRT and NRTIs, followed by NRTI, and few of them against the protease inhibitors. Uh, from the genomic signatures, we know that the resistance mutations were sequestered in the regions which has high genetic variability or mutation rate, suggesting that these are unstable areas and might change further in future, which give rise to newer mutations. Uh, and the concerning point I showed you is that the patterns of genomic variability, especially in the sequences of the naive patient, they resemble more of the virological failure patient. So, which is really concerning because these patients haven't started the therapy yet. And finally, the analysis of the data from many studies and sequences throughout the country, from different regions of the country, is required to truly understand the nature of subtype diversity, the relationship between epidemics within the country, and to truly drive the relationship between genetic variability and occurrence of drug resistance mutations. So I would end my talk by acknowledging many people who make these kind of research as possible. Dr. Syed Ali from Nazarbayev University, Dr. Faisal Mahmood from AKU, Dr. Fatma Mir from AKU, Dr. Rashida Farhan from London School of Hygiene and Tropical Medicine, Dr. Sharaf Ali Shah from Bridge Foundation, our National and Provincial AIDS Control Program, Professor Sarah Loren Jones from University of Oxford, and Dr. Joachim Ipsonsion, who have University of Lund, who have contributed widely in these projects, especially Dr. Faisal and Dr. Fatma and Dr. Sharaf Shah, without whom the most of the researches would not have been possible. Also, I would like to acknowledge my research students, Khalid Ahmed, Ursa Tarek, Ramsha Khalid, Dilsha Siddiqui, Nida Faruqi, Ali Salman Sheikh, Fiza Nazim, and Shabnam Khan, who have worked extensively and made many of these studies possible. So most of them have graduated or about to graduate, but is there these students make vital contributions for the understanding of HIV in our country. We would also like to acknowledge the funding agencies, Higher Education Commission Pakistan, Pakistan Science Foundation, World Health Organization, HIV Research Trust, International Society for Infectious Disease, and European Society for Clinical Microbiology and Infectious Disease for funding most of the research and the data that have been generated through their generous support. So with this, I would like to thank you so much for your attention, for bearing with me and listening to this. There's a lot of work to be done and an overwhelming amount of facts. So especially with respect to our country, if there are any questions, I'll be happy to answer. Thank you very much.